Hey everyone, in this video lecture, we are going to cover the topic of dihybrid crosses. So review and recap here from our last video. Our last video talked about inheritance of one single gene. And so during meiosis, we looked at how each haploid gamete, which is either a sperm or an egg, randomly inherits only one copy of each allele. So in this example here, the diploid cell might have the genotype big P, little p. So the haploid gametes potentially could have the big P allele, or they could have a little p allele. So when it goes to creating or predicting the inheritance of this trait for flower color, we can use a simple 2x2 Punnett square to predict the trait of the offspring. We take the two alleles of the parent and we separate them. So if this is the egg on the left side here, I separate the big P and little p of the parent. If this is the father here, we have big P, little p separated in the two different sperm cells. And then in each box, we fertilize these sex cells by combining the egg and the sperm to create the offspring's genotype. Well, this is nice and simple because we're only dealing with one gene at a time. That is not always the case, though, because there might be cases where we're inheriting two genes that are linked together. So we want to look, if we're looking at the inheritance of two genes that are on different chromosomes, we need to look at all possible combinations of the alleles in those gametes. So it's pretty simple before. If you only had two alleles, you know that some of the offspring are going to have one allele, some of the offspring or some of the sex cells will have the other allele. Nice and simple. But when we look at two different genes, each on different chromosomes, we need to look at every possible combination of the alleles in the haploid gametes. So if this diploid cell has a chromosome for this P gene, and it has the genotype big P, little p. It has a second set of chromosomes with this gene that we're representing with the letter T, and this individual has the big T, little t genotype. During meiosis, it is possible that we create a sex cell that has both big P and big T alleles. It is also possible to create big P, little t alleles. It is possible to create a sex cell with the big P, little t allele. And lastly, it is possible to create a sex cell with the little p, big T allele. So because each sex cell is possible, when we go to create a Punnett square to predict the traits of these offspring, we need to look at every possible allele combination. And this is really looking at both the law of segregation and the law of independent assortment when we go to create this Punnett square. And so in order to help us predict the inheritance of these two genes that are not on the same chromosome, we can use a simple method to come up with every possible allele combination. So again, if we're looking at one of Mendel's pea plants, the genotype, because we're looking at two genes, is now going to be represented by 4 letters. So the first 2 letters represent the first gene, which is flower color. So this individual is big P, little p for its genotype. The second gene is big T, little t. And so again, the whole genotype is now 4 letters. And the phenotype here is that we have purple flowers and the plant is tall. So we have 2 phenotypes to represent. If I take that genotype, here again are all the possible combinations of the alleles for each trait that this individual would create during meiosis. So how do we actually predict, or really kind of simply, quickly come up with the different allele combinations? So if we have a situation where we don't have all dominant alleles or all recessive alleles, and we get this kind of combination of heterozygous combinations, then we need to come up with a simple method for predicting allele combinations. And for that, we use what we call the FOIL method. FOIL meaning the same FOIL that you think of in algebra class. FOIL is an acronym that stands for first, outer, inner, last. And this is how it's basically a shortcut for coming up with every possible allele combination that might be created during meiosis. And so if I take that same genotype, the big P, big T, big little t, we have 4 letters. The first 2 letters are for gene 1, second 2 letters are for gene 2. I want to look at the allele combinations that are created in my sex cells. So for first, I'm going to take the first allele of each trait. I'm going to take the first of the P trait, first of the T trait, and combine them together. So the first here is a big P and a big T. I'm going to write them next to each other. O stands for outer. I'm going to take the outside 2 alleles in this 4-letter genotype. So I'm going to take the outside P and the outside T, and I'm going to combine them together, representing the alleles of big P and little t. I stands for inner or inside. I'm going to take the inner 2 most alleles, which is little p, big T. Note how I typically like to write uppercase letters first. In this case, I do not want to do that. I want to keep all the P alleles first and all the T alleles second. And then the L in FOIL stands for last. So I'm going to take the last of the P trait and the last of the T trait, and I'm going to combine those 2 together to have little p, little t. So this represents every possible allele combination between G1 and gene 2. So first step when we are trying to predict the offspring in a dihybrid cross where we're looking at 2 separate traits at the same time. Step 1 is make sure that you know both the phenotype and genotype of the 2 parents that are being mated together. So in this case, both parents, if we're thinking about Mendel's pea plants here, both parents have a phenotype of green and round peas. Their genotype is both heterozygous, so big G, big R, little r. So the first trait here, big G, is for the color of the pea, and then the second trait here, the R, is representing the shape of the pea plant. Step 2 is we're going to use the FOIL method, first, outer, inner, last. That is going to come up with all the possible combinations of alleles between these 2 different genes in each parent. So you make sure you FOIL for parent 1, FOIL for parent 2. Because both parents have the same genotype, you really only have to FOIL once here. So the F would be big G, big R. The O is big G, little r. I is little g, big R. L is little g, little r. Step 3, you're going to take the foils for each of the parents and you're going to put them across the top and down the side of your Punnett square, just like you would do in a regular smaller Punnett square. You put those combinations, or you put the alleles across the top and down the side. So I put parent 1 across the top here. I put the FOIL combinations of parent 2 down the side. Then we fill in just like you'd be filling in for a regular small Punnett square of 1 trait. I'm going to start by doing trait number 1, the G or color of the peas first. So I'm going to take the big G from this parent, combine it with the big G of this parent, and write that in the box. And I'm going to repeat this across every single box. You'll notice down here in this last one, I have a little g from this parent, a little g from parent 2, combining together to give me the genotype for color, little g, little g. Step 5 is you're now going to repeat this, but for the second trait only. So now I'm going to go through and fill in all the R's based on the combination of the 2 parent genes or alleles. And then step 6 is identify the phenotypes of these potential offspring. So in this case, the dominant version of the G trait is green. The recessive version of the green trait is yellow. The dominant version of the R trait is round. The recessive version of the R trait is wrinkled. So anywhere we see big G's and big R's, they're green and round. In this one here, we have big G for green, but we have little r's, so that means it's going to be wrinkled in shape. Over here, we have recessive in color, so we have the yellow color, but we're dominant for shape, so it's round. This is the only one that is fully recessive. It is little, it's recessive for color, so it's yellow, and recessive for shape, so it is wrinkled. And so lastly, we are going to look at the ratio of the different traits. So because we're dihybrid, we're looking at 2 traits at the same time. We need to count up not how many are green. We want to look at how many are both green and round in shape. So if I count the number that I see that are green and round, I see 9 of them. If I want to then count how many are both green but wrinkled, I have 3 of those. I have 3 that are yellow and round, and then I only have 1 that is yellow and wrinkled. Now, this ratio of 9:3:3:1 is typical if you have 2 parents, both heterozygous for both traits. You will see this 9:3:3:1 ratio come up frequently when both parents have both fully heterozygous genotypes. So the big G, little r, little r.